So far, we have discussed how glucose is consumed. However, every metabolic interconversion is a road in two directions. If glucose is consumed somehow it must be built somehow. For example, if you take photosynthetic organisms, all light energy at the end is harvested in the form of carbohydrates using atmospheric CO2 in the process which is called Calvin cycle. And in this process, the glucose is produced as intermediate and then used to build various carbohydrates. Likewise, in animals and specifically in our body, if the glucose supplies are over both dietary and intracellular supplies in the form of glycogen, so the glucose needs to be regenerated, produced de novo somehow to maintain glucose homeostasis. Specifically because some of our tissues are highly dependent on glucose. If you take brain tissue, erythrocytes, renal medulla, or embryonic tissues, they need constant glucose supply. It needs to be produced somehow. And in different organisms in our body, including our body, there is a versatile system to produce glucose from various precursor molecules. And collectively, this pathway where various precursors feed to produce glucose is called gluconeogenesis pathway, sorry. So in fact, the gluconeogenesis pathway is almost the same as glycolysis pathway. It shares 7 out of 10 enzymes, 7 out of 10 reactions with glycolysis. And the question how these two pathways can live together. When we talked about glycolysis, we have learned that glycolysis, although it produces ATP, is a highly exergonic process. A lot of free energy is released. So to answer these questions, we shall look back at the energetics of glycolysis pathway. If we would measure free energy of different steps of glycolysis energy drop, then we would see that 7 out of 10 pathways have energy drop around 0. They are fully reversible. And in fact, these pathways are shared, these steps are shared between glycolysis and gluconeogenesis. They are catalyzed by same enzymes and these are the same reactions. However, there are 3 steps which allow glycolysis to occur in one direction where energy drop is highly negative. And these steps are different in glyconeogenesis pathway. These are called bypass steps. So they are catalyzed by separate sets of enzymes and they're separately regulated. So when in the next few slides we will look at these bypass steps one by one, starting with the lowest point in the gluconeogenesis pathway, starting from pyruvate. Of note, so if we consider bypass steps, the tissues like muscle tissue or brain tissue, which are totally dependent on glycolysis, they do not have this high activity of gluconeogenesis enzymes. However, tissues like liver, renal cortex, or the epithelial cells of small intestine, which can produce de novo glucose. So they run this gluconeogenesis pathway. They have this enzyme catalyzing bypass steps. So the first part of first bypass is the conversion of getting from pyruvate to entry into glycogenesis pathway. Remember, pyruvate is the low energetic molecule. And the first thing which happened in the first bypass is to produce a molecule called oxaloacetate from pyruvate. And this is done by an enzyme called pyruvate carboxylase, which uses energy of ATP and cofactor called biotin to attach covalently attach activated bicarbonate anion to the third residue of pyruvate. Of note, this reaction is happening in mitochondria. Pyruvate can get transported through mitochondrial membrane and get into mitochondria. So we'll see later why this compartmentalization is necessary. But if you consider the pyruvate decarboxylase, carboxylase, it is an interesting enzyme consisting of having two sites. As mentioned, it has this cofactor biotin. And biotin is also known as vitamin B7. It is covalently attached to lysine residue in pyruvate decarboxylase, and it is used as an acceptor for activated carbanion. It covalently binds carbanion using the energy of ATP. In a sense, function of biotin is analogous to the function of thiamine pyrophosphate, which we have discussed in previous part, which is in the case of thiamine pyrophosphate, it is carrier of active aldehyde group. So biotin is a carrier of active carboxy group. This activated carboxy group travels to second site in pyruvate decarboxylase, and then it is loaded covalently on pyruvate to produce oxaloacetate. So this step happens again in mitochondria. However, then oxaloacetate travels back to cytosol. We'll see later how it happens and why it happens. But in the cytosol, it is converted to phosphoenol pyruvate, which originates the backwards direction in the gluconeogenic pathway, and it converted by means of an enzyme called phosphoenol pyruvate carboxykinase. It is using another energy ATP equivalent, but in form not form of ATP, but in form of GTP, which is energetically the same, to unload back the carboxy residue with concomitant phosphorylation. So one could note that these two reactions, the production of oxaloacetate and by loading carboxy group, and next production of phosphoenol pyruvate by unloading carboxy group, it is the same carboxy group loaded and unloaded. So the question why it is needed, and the reason is that there is a huge free energy difference between the phosphoenol pyruvate and pyruvate. So phosphoenol pyruvate is much higher, much more energetic molecule, and to load the necessary energy into pyruvate, one ATP equivalent is not sufficient. So loading and unloading of carboxy group, it is a step of activation which conserve energy, splitting this energetics necessary for this conversion into two steps, because there is no ATP like equivalent which would be sufficient to pump whole necessary energy into phosphoenol pyruvate. So this is used not only in this reaction but in many other reactions So next question, why this compartmentalization shuttling is necessary and how it's happening? To understand it, so we need to consider the fact that during the gluconeogenesis we need this reducing equivalence in the form of an ADH in the cytosol, because if you remember from the glycolysis, so when going from glucose to pyruvate, at one step we produce reducing equivalence. In the case of glyconeogenesis, the amounts of NADH in the cytosol, and generally amounts of NADH in the cytosol, are low. So on the other hand, in mitochondria, the concentrations of NADH and other reducing equivalents are much higher, because tricarbonic acid cycle constantly grinds everything into the form of reducing equivalents to feed electron transport chain. So as a matter of fact, the steady state concentration difference of NADH between cytosol and mitochondrial matrix is approximately 4 orders of magnitude, 4 or 5 orders of magnitude. So and this fact is used at the end to feed the supply NADH into the cytosol. So the way it happens that oxaloacetate produced in mitochondrial matrix is converted to malate by reducing oxaloacetate by NADH. So there are channels in the mitochondrial membrane which will transport system in the mitochondrial membrane, which is allowing to exit mitochondria back into cytosol, and malate is exported in the cytosol. So in mitochondria, in the cytosol, so there is analogous enzyme as compared to mitochondria, which is called malate dehydrogenase. So both mitochondrial enzymes and cytosolic enzymes are very similar, so they perform the same operation. But cytosolic malate dehydrogenase unloads back NADH from malate converted to oxaloacetate. So this way, we start with pyruvate and end up with oxaloacetate and reducing agents which are practical for gluconeogenesis pathway. Then this oxaloacetate is in the cytosol again converted by PEP carboxykinase into phosphoenol pyruvate. So in fact, there is another flavor of this pathway, which does not require when the transfer of an ADH in the cytosol is not required. This happens, for example, in the liver when gluconeogenesis is ran from lactate. When excessive lactate is produced by, let's say, by lactate fermentation muscle, it at the end ends up in liver, and liver uses lactate to produce to run through gluconeogenic pathway. In this case, the lactate can be converted to pyruvate in the cytosol by lactate dehydrogenase. In this case, NADH is already produced. In this case, you don't need this malate shuttling to get NADH. In this case, again, pyruvate travels to the mitochondria. It is converted to oxaloacetate by the same way by pyruvate carboxylase, but in this case, oxaloacetate straight converted to phosphoenol pyruvate by mitochondrial flavor of PEP carboxylase, and PEP can travel through the mitochondrial membrane similar to pyruvate. So this way, PEP ends up in back in the cytosol. So this way, in both cases, you end up with NADH and phosphoenol pyruvate, which are necessary to fuel gluconeogenesis pathway, but in a slightly different way. And balance between these two flavors of first bypass, it is dictated by the availability of lactate. So in the second bypass, so before second bypass, so there is this series of reactions, and in this series of reactions of note, two things happen before the second bypass. First, two more ATP molecules are consumed. So if we go back, recap the first bypass. So for each glucose molecule to produce one glucose molecule, we need to start with two phosphoenol pyruvates. So we need to convert two pyruvates to two phosphoenol pyruvates. For one phosphoenol, to produce one phosphoenol pyruvate, we need to two ATP equivalents, one ATP and one GTP. So for two ones, we need four molecules. So then once phosphoenol pyruvate travels backwards in the gluconeogenesis pathway, so there is a step which is opposite of converting 1,3-bisphosphoglycerate to 3-bisphosphoglycerate. So it needs to consume two more ATP molecules in total, because this step is opposite of substrate level phosphorylation, which happens in glycolysis. And then you would need to consume two NADH reducing equivalent to get to glyceraldehyde 3-phosphate. So then finally, through this opposite rebuilding 6-radiocarbon backbone, we end up with fructose 1,6-bisphosphate. In this step, if we go back to glycolysis pathway, we would remember that this is a highly energetic molecule. It was at the end produced from glucose in two steps by two ATP-dependent phosphorylation. So in principle, this could be run from the energetics perspective, from thermodynamics perspective. It can be hydrolyzed freely with good energy drop back into fructose 6-phosphate. But to hydrolyze it back, it is using an enzyme which is different from glycolysis pathways. If in glycolysis pathway, this reaction is catalyzed, forward reaction catalyzed by phosphofructokinase, then in gluconeogenesis, the backward reaction is catalyzed by fructose 1,6-bis phosphatase, and the reason is that one would like to avoid futyl cycles. So if we would have this reaction catalyzed by the same enzyme, so then this enzyme would this step would essentially act as ATP consumption step, which would be constantly consuming ATP. So because of that, these two enzymes are completely different, and they are completely different regulated, and in fact regulated in a reciprocal manner, allowing to separate these two pathways. Likewise, glucose 6-phosphate is more energetic molecule than glucose, and because of that, the third bypass step is catalyzed by different enzyme. If in glycolysis the forward reaction is catalyzed by hexokinase, so then in gluconeogenesis, so backward reaction is catalyzed by glucose 6-phosphatase. And again, these are two reciprocally to differently regulated enzymes, which avoids the futyl cycles. Glucose 6-phosphatase is not expressed in the tissues which do not do gluconeogenesis, like in brain or muscle tissue, for a simple reason, because if you want to keep glucose in a cell, you do not want to dephosphorylate it. only if you want to export it. So it is confined to gluconeogenic tissues, mostly to liver. In the liver, this enzyme is located in the lumen, in ER lumen, and it's a final step in gluconeogenesis pathway. So in total, if you look at this ins and out of gluconeogenesis, for one glucose molecule, we would need to take 2 pyruvates and in total 6 nucleotide 3-phosphate equivalents and 2 reducing equivalents in the form of an ADH. So in total, this is a highly uh, energetically inefficient step, but again, it has its own essential role in ensuring glucose homeostasis. So there are various ways how the gluconeogenesis pathway can be fed. In biological systems, the gluconeogenesis is extremely versatile. For example, in plants or in some microbes like yeast, the fatty acids could be fed into gluconeogenesis by starting with acetyl-CoA in mitochondria. However, this is not the case in our cells. In our organisms, we cannot convert fatty acids back into glucose.